Hello everyone. Today we'll be talking about the structure of proteins. These are the objectives. Proteins have a unique three-dimensional shape. It is this unique 3D shape of a protein that determines its function. Now, whether a protein like keratin provides structure to the hair and nails, or whether a particular protein acts like an enzyme, or whether it acts like a mobility protein like actin or myosin, or whether it acts as a transport protein in the form of hemoglobin that transports oxygen molecules throughout the body, it all depends on its particular structure. Amino acids just don't kind of come together and hope that they make the right protein. There is something very specific called the primary structure, which dictates a specific order of amino acids in a protein. The primary structure of a protein is simply the list of amino acids in the protein from the start of the protein, called the N-terminus that has the free amino group, to the end of the protein called the C, or the carboxy terminus, that has the free carboxylic acid group. The most common types of secondary structures are the alpha helix and the beta pleated sheet. The secondary structures are stabilized by hydrogen bonds, which form between the oxygen of the carbonyl group of one amino acid and the hydrogen of the amino group of the second amino acid. Next is the tertiary structure, which is the three-dimensional structure of a protein. Secondary structures such as alpha helices and beta sheets fold on themselves to form the tertiary structure of the protein. The protein quaternary structure is the number and arrangement of multiple folded protein subunits in a multi-subunit complex. For example, you have hemoglobin, which is a globular protein that has a quaternary structure. The hemoglobin molecule is made up of two alpha chains and two beta chains. This 4 polypeptide chain molecule is bound to each other by disulfide bonds, hydrogen bonds, hydrophobic interactions, which we'll talk about later. So, what is the native conformation of a protein? For a protein to be functional, it needs to be in its native state, which is called the folded state, where the protein has arranged itself into a neat, compact shape with the help of these four bonds we talked about: disulfide bonds, hydrogen bonds, ionic bonds, and hydrophobic interactions. When some of these bonds are broken with the application of heat, for example, a change in pH, or by the addition of a chemical, the protein denatures and returns to its primary configuration. That is called the denatured or the unfolded state of a protein. A denatured protein is no longer capable of carrying out any function. So let's see what the primary structure of a protein looks like. So here you have valine and here you have alanine coming together. So the carboxyl group of the valine and the amino group of alanine react together, lose a water molecule through a reaction called dehydration synthesis, form the peptide bond, and here we have our dipeptide. Okay, a dipeptide is a peptide of two amino acids, valine and alanine. So now on this dipeptide, which technically is a protein because it is more than one amino acid, on this dipeptide you have the free end, which is the starting end, which is the amino terminal, and then you have the terminal end or the end of the protein, which is the carboxyl terminus. So every protein, irrespective of how short or long it is always going to have the amino terminus as its starting point and the carboxyl terminus as its ending point. So these are abbreviated as the N terminal and the C terminal. Now let's talk a little more about the peptide bond. The peptide bond between the carbon and nitrogen is a pretty strong bond and is not broken by conditions that denature proteins such as heating. In order to break the peptide bond, you need to have a prolonged exposure to a strong acid or base at elevated temperatures and for a really long time, sometimes for as long as up to 24 hours. To hydrolyze these bonds, non-enzymatically. So for example, when you boil an egg, you are denaturing the protein, which means you are simply breaking down the secondary and tertiary structure, but the protein is still retaining the primary structure, as heating is not sufficient to break the peptide bonds between the individual amino acids. Let's take a look at the characteristics of the peptide bond. The peptide bond is said to have a partial double bond character. Now, what does that mean? The bond between carbon and nitrogen is a single bond. Okay. In spite of the fact that it is a single bond, it behaves like a double bond. Okay. One advantage of this is the side chains can exist in a trans configuration, which causes less structural hindrance as compared to the cis configuration. So here you have two amino acids coming together, and now they have their side chain in trans configuration. Here you have two amino acids coming together. They have their side chains in cis configuration. You are more likely to find trans configuration as compared to cis configuration because these two bulky side chains sitting right next to each other causes a structural hindrance, whereas here they are separated apart, which makes them less likely to hinder each other. So in peptide bonds, the vast majority take the trans conformation, with typically the ratio being you'll find one cis configuration to 1,000 trans configurations. So this preference for trans configuration over the cis orientation is because there is less structural hindrance. Now, for the next few slides, what we are really going to talk about is how do you analyze the protein sequence. And why is this important? Because recombinant DNA technology, which is a technology that is used to make recombinant proteins, is a technique that is used to make a lot of medicines. For example, you have insulin, which is, as you all know, is a medicine that is used to treat diabetes. This insulin is a recombinant protein that is made using recombinant DNA technology. So when you have synthesized this protein, insulin, you need to be able to correctly identify the exact amino acid sequence of that particular protein, because as by now you know, it is the structure of the protein which determines its function, and it is the sequence of the protein, as you will see shortly, that determines its structure. So it's important to have the correct amino acid sequence. Another example of a recombinant protein is erythropoietin, which is a recombinant protein that is used to treat anemia. So primarily there are these three steps, or rather these three techniques that are used to identify a protein sequence. The first one is the amino acid composition. The second one is Edman degradation, and the third one is the cleavage of polypeptide. So the protein sequence analysis using amino acid composition is one of the very primitive techniques. It is no longer really used in practice, and we'll see why. But at the same time, it was one of the first techniques that was used to determine the amino acid composition in a protein. So what really happens here is the protein, or which is also called a peptide. So if I use these words interchangeably, don't get confused. The peptide is hydrolyzed using a strong acid like hydrochloric acid at a high temperature for as long as up to 24 hours, which breaks the peptide bonds and separates the amino acids. So at this point, what you really have now is a mixture of various amino acids that make up that particular protein in solution. Next, ion exchange chromatography, which is a fairly complex technique, is used to separate these charged amino acids. So from the last lecture, you know that amino acids that make up the proteins are zwitterionic compounds that contain both positively and negatively charged functional groups. Depending on the pH of the environment, the proteins or the amino acids may carry a net positive charge, a net negative charge, or they might be neutral. So you can theoretically predict using the pKa the elution sequence of amino acids for a particular buffer and a particular type of ion exchange column. There are two types of ion exchange columns primarily. You have a cation exchange column that separates out the positively charged molecules, and you have an anion exchange column that separates out the negatively charged molecules. Without getting too much into the specifics, for example, how this thing works is you have aspartic acid that has the lowest isoelectric point of 2.7, and then you have arginine that has the highest isoelectric point of 10.7. So if you use a particular buffer, for example, in this case it's zero point 0.2 molar sodium nitrate, you can predict that aspartic acid will be the one that will be eluted first and arginine will be the one that will be eluted last. Okay. So ninhydrin, which is originally a yellow powder, is used to react with these amino acids. And when ninhydrin reacts with the amino terminus of each of the amino acid, it turns a deep purple color. It is this deep purple color that is detected in this method in terms of absorbance. So the more the number of amino acids present in a protein, the higher the absorbance. For example, in this case, you have the absorbance for glycine is twice as much as the absorbance of aspartic acid, alanine, phenylalanine, and arginine, which means that this particular peptide is made up of two glycine molecules or two glycines and one each of aspartic acid, alanine, phenylalanine, and arginine. One big drawback of this method is what it really does is it tells you this particular peptide is composed of these specific amino acids. What it does not tell you is the sequence of these amino acids, which is extremely important piece of information, but we don't really get that using this simple primitive technique, and that is the reason this particular technique is no longer in practice. So what has replaced the amino acid composition is Edman degradation, named after the person, the scientist who invented this method. So in Edman degradation, what it really does is it sequentially removes one amino acid residue at a time from the N terminus of the peptide. A chemical called phenyl isothiocyanate, abbreviated as PTH, is used to react with the N-terminal amino acid under mildly basic conditions. So what it does is it forms this derivative, a cyclic derivative of that particular amino acid, which is then liberated or which is broken off using mildly acidic conditions. So it really breaks off that particular derivative of phenyl isothiocyanate. So in this case, you have the first amino acid, which is alanine. PTH is reacted with alanine that forms this derivative of PTH. In using acidic conditions, this derivative is cleaved off. So now you have alanine getting separated, which now makes lysine, which is the second amino acid, the first amino acid in the shortened peptide. So this particular sequence is repeated for the number of amino acids that you have in a particular peptide. So in this peptide, which has 5 amino acids, you would have to repeat this reaction 5 times. So each time you have a derivative of PTH, which is forming a complex with that particular amino acid that is getting broken off. And then this particular derivative is then identified using the chromatographic procedure that we just discussed earlier. So while the amino acid composition by itself is not used any longer to identify the amino acid composition of a protein, it is still used in the context of identifying the individual amino acids. And then the sequence is repeated until you are done with all the amino acids. The next one is what is called the enzymatic cleavage of polypeptide. Now, one of the limitations of Edman degradation is most of the proteins are 1,000 amino acids long. So it really makes no sense, at least no practical sense, in repeating this reaction 1,000 times, changing the pHs from basic to acidic conditions 1,000 times. So what really is effective is combining the Edman degradation with the enzymatic cleavage of polypeptide. Now you can imagine a polypeptide that's 1,000 amino acids long. What really happens in this enzymatic cleavage is there are certain enzymes which cleave amino acids, which cleave particular amino acids, I should add. For example, there is this enzyme called trypsin that is going to cleave or cut the particular peptide after lysine residues and after arginine residues. It's going to leave everything else untouched. So anytime it encounters a lysine or a arginine in a peptide sequence, it is going to cut it off. So likewise, you have another enzyme called cyanogen bromide that specifically cleaves only after methionine. You have another enzyme called chymotrypsin that specifically cleaves only after the aromatic amino acids, which are, there are three amino acids which are aromatic because they have a benzene ring in them: tyrosine, phenylalanine, and threonine. So this particular enzyme is only going to cleave after these three amino acids. So the way this technique is used is you have a peptide of unknown sequence. So first you cleave this peptide using one enzyme. In this case it's trypsin, which is only going to cleave after lysine and arginine. So then you are going to end up with some fragments of that particular peptide. In this case it's peptide A, B, C. Now the question really is what is the correct order of these peptides? Is it ABC? Is it ACB, BAC? That you don't really know at this point. So in order to get that information, the same protein is now cleaved with a different enzyme. In this case, it's cyanogen bromide. It could be really anything. It could be cyanogen bromide, it could be chymotrypsin, but you cleave the same peptide with the second enzyme, and what the second enzyme does is it is going to cleave after its own specific amino acid. So now you have a particular order of amino acids which can be used to derive the correct sequence. Is it ABC, ACB, or BAC? Let's take a look at an example. So I have a particular peptide that I have digested using trypsin. So the ones in pink that you see here are the fragments that I have obtained using trypsin. So you'll see that it has cleaved after lysine. It has another fragment which it has cleaved after lysine. And then the smaller fragments, you can identify their sequence using Edman degradation, because now it's more realistic to use Edman degradation to identify this 5 amino acid or 4 amino acid sequence. But at this point of time, I still don't have the information as to what exact sequence this particular peptide has. So what I'll do next is I'll use the same peptide, and this time I'll digest it using a different enzyme, chymotrypsin. So chymotrypsin, because it cleaves on different amino acids, in this case it's aromatic, so this particular peptide has just one aromatic amino acid, which is tryptophan. So it's going to give me this cleaved fragment. It has cleaved after tryptophan. So now I have this information. I have these two tryptic peptides coming from trypsin. I have this one peptide coming from chymotrypsin. So what this really does is, what it tells me is the particular amino peptide that we are talking about has this particular amino acid sequence. It's just about putting a jigsaw puzzle together. So I have valine, lysine, followed by alanine, alanine, tryptophan, which in this case I'll get that sequence if I put this particular peptide right after this one. So then I have valine, lysine, alanine, alanine. So using the chymotryptic overlap peptide, I've been able to put together the sequence, the correct sequence of my particular peptide. This is how the enzymatic cleavage of polypeptide works in conjunction with Edman degradation. So here's an example. So you might be asked on the test to identify the peptide fragments that could result from the cleavage of a peptide using trypsin and chymotrypsin. So it's really important for you to know that the particular amino acids that these particular enzymes cleave after. So you have trypsin, cleaving after lysine and arginine. You have chymotrypsin cleaving after the aromatic amino acids, and then you have cyanogen bromide cleaving after methionine. And this is where really the one letter codes for amino acids become important. So now let's talk about secondary structure. The secondary structure refers to structures that form within a polypeptide due to interactions between atoms of the backbone, which brings us to the question, what is a peptide backbone? The backbone just refers to the polypeptide chain apart from the R groups. So all of this is the peptide backbone. The side chains on the amino acids are not a part of the peptide backbone. The most common types of secondary structures are the alpha helix and the beta sheet. Both these alpha helices and beta sheets are held in shape by hydrogen bonds which form between the oxygen of carbonyl of one amino acid and the hydrogen of the amino group of the second amino acid. In alpha helix, and this is how it really works, and we have a better picture of this. You have the oxygen of the carbonyl of one amino acid forming a hydrogen bond with the hydrogen of the amino group of second amino acid and holding the structure in place. So you have these hydrogen bonds all across the peptide backbone that hold together the alpha helix and beta sheets. Now there are certain amino acids that cannot be a part of alpha helices, and one such amino acid is proline. Now this is the structure of proline. Now what makes proline unique is proline is the only amino acid that has its R group bending upon the backbone. So if you consider this to be the backbone of the amino acid, you have the R group, which is this hydrocarbon group, which is forming a bond with the backbone. So what it really does is, now this particular backbone does not have a free hydrogen for it to participate in hydrogen bonding. That makes proline an unlikely amino acid to feature in an alpha helix. So the way this hydrogen bond is formed is very specific. It is formed between every n plus 4 amino acid, which means the first amino acid is going to form a hydrogen bond with the fifth. The second amino acid is going to form a hydrogen bond with the sixth, and so on. And this picture is not the exact good representation of how this takes place, so let's take a look at this picture. Here's your first amino acid, second amino acid, third, fourth, and so on. Each of them is numbered. And here you have your oxygen of the carbonyl group of second amino acid forming a hydrogen bond with the hydrogen of amino group of sixth amino acid. Oxygen of the carbonyl group of first amino acid forming a hydrogen bond with the hydrogen of amino group of fifth amino acid. So this hydrogen bonding happens throughout the secondary structure, and it happens in a very specific manner. Which brings us to the second part of the secondary structure, one of them being alpha helix. The second one is beta sheets. In a beta sheet, two or more segments of a polypeptide chain line up next to each other, forming a sheet-like structure that is again held together by hydrogen bonds. The hydrogen bonds form between the carbonyl and amino groups of the backbone, while the R groups extend above and below the plane of the sheet. For example, here you have two beta sheets lying next to each other, and then you have the R groups extending above, or rather below and above the plane. And then you have the hydrogen bonds forming between the carbonyl group of one amino acid on one sheet with the hydrogen bond of the second amino acid of the second sheet. So the strands of a beta plated sheet may be parallel, in which case they are pointing in the same direction, meaning that their N and C terminus match up. Or they can be anti-parallel, in which case their N and C terminus are going in the opposite directions. So you can have anti-parallel beta sheets and you can have parallel beta sheets. Now, in addition to the regions of repeating secondary structures like alpha helices and beta sheets, Protein also contain secondary structures of non-repetitive regions. These sequences are called loops or turns. These loops or turns, typically also called beta bends, have a definite purpose. What they do is they allow the protein chain to fold back on itself to produce the compact shape associated with most globular proteins. So you'll see that these beta turns are allowing the sheets to fold right next to each other instead of just extending in one long direction. So you'll have these turns and loops connecting two alpha helices or two beta sheets. And they are called non-repetitive secondary structures because they don't repeat as regularly as your alpha helices and beta sheets. So what are supersecondary structures? If we think of each secondary structure as a unit, that is, example, one helix would be one unit, one beta strand would be one unit, then a supersecondary structure is typically composed of at least two units of a secondary structure. For example, it could be beta alpha beta, alpha beta alpha. You can have two alpha beta, or you can have more. So anytime you have more than two secondary structures together, it's called a super secondary structure. The tertiary structure. The tertiary structure is the overall three-dimensional structure of a polypeptide. The tertiary structure comes about primarily due to the interactions between the R groups of amino acids that make up the protein. So bear in mind the distinction that secondary structure is stabilized by the hydrogen bonds along the backbone of the peptide, in which the R groups have no role to play whatsoever. The interactions between the R groups or the side chains of amino acids are what make up the tertiary structure of a protein. So these R group interactions that we'll talk about in a second include disulfide bonds, hydrogen bonds, ionic bonds, and hydrophobic interactions. So what is a domain? A protein domain is a part of a given protein sequence and tertiary structure that can evolve, function, and exist independently of rest of the protein chain. So essentially, if you consider this particular protein that has four domains, each domain has a specific name, a specific function. For example, the px domain is involved in binding. The pH domain is involved in subcellular targeting. The PLD domain has enzymatic activity. So each of these four domains can exist without the presence of the other. So the px domain, if you were to cut this protein off at this particular point, the px domain would still be functional. It is independent of all of the other domains. So you can think about a domain as sort of a functional protein within a protein. Okay, but it can exist without the rest of the structure of protein. The pH domain can be isolated from this protein and it can still carry out its function. So a domain can carry out its function and it can fold into a compact three-dimensional structure without the existence of other parts of the protein. Now, if you consider this white sequence right in the center between pH and PLD domains, that is not a domain. So for this particular structure to fold, it would need to have the existence of all of the other parts of the protein. Now, whether that particular region has any significant biological function, we wouldn't know. But so that is the difference between a domain and other regions of the protein. A domain can exist, function, fold independently of the rest of the protein. That brings us to interactions that stabilize the tertiary structure. Disulfide bond. A disulfide bond is a type of a covalent bond that contributes to stabilizing the tertiary structure. Disulfide bonds are covalent bonds between sulfur-containing side chains of cysteines. Now remember that there are only two amino acids which contain sulfur as a part of their side chain. One of them is methionine, the other one is cysteine. However, methionine cannot contribute to the formation of disulfide bond. Only cysteines can contribute to the formation of a disulfide bond. So here you have two cysteines along the polypeptide backbone. They are in a reduced environment because they have hydrogen. Now, if you were to move this particular protein into an oxidizing environment, for example, a region where there is some presence of oxygen, the disulfide bonds will undergo oxidation. They will lose the hydrogen and they'll form a bond between the two sulfurs of the two cysteine residues. So you can think about these disulfide bonds as molecular safety pins that keep the part of polypeptides firmly attached to one another. Next, we have hydrophobic interactions. So here you have two amino acids, isoleucine, leucine, which are along the polypeptide backbone. And what really hydrophobic interactions are all about, you'll notice that both these side chains have non-polar side chains. So hydrophobic interactions are more entropy driven in the sense that these hydrophobic groups are sitting close to each other, not as much for their like for each other, but because they dislike water. They repel away from water and are therefore closely stacked to each other, which adds to the structural stability of the protein. Then we have hydrogen bond, which we have spent ample time talking about, but just to summarize it, hydrogen bonds are formed between hydrogen and an electronegative atom, which in this case is oxygen in the case of amino acids. And lastly, you have the ionic bond. R groups with like charges repel each other, and R groups with opposite charges can form an ionic bond. So here you have aspartate with carboxyl group carrying a negative charge. You have lysine carrying the amino group, which is protonated and therefore carries a positive charge. So in this case, the deprotonated carboxyl group can form an ionic bond with the protonated amino group. Protein folding, unfolding, and altered folding, or it can be also referred to as misfolding. It is important for a protein to be folded into a compact series shape in order for it to be functional. Now, what really are chaperone proteins? Many proteins can't fold by themselves and need some help. This help comes in the form of chaperone proteins, which are specialized proteins which help other proteins get into a 3D folded shape. Now, the way this protein folding happens is in a very specific manner. As a protein is getting synthesized in the ribosome, it doesn't really wait until all of the protein is synthesized for it to start folding. As soon as a part of the protein gets synthesized, it starts folding even as the remainder of the protein is still getting synthesized. And it happens in specific steps. So it starts off by forming secondary structure, then it starts off by forming domains, if any. It's not necessary that every protein have a domain, and then the protein finally folds itself into a tertiary folded shape. And it is important for the protein to be in this folded native state in order for it to be functional. Denaturation. So if the temperature or pH of a protein's environment is changed, or if it is exposed to chemicals, these interactions that stabilize the protein tertiary structure can be disrupted, causing the protein to lose its 3D structure and turn back into an unstructured string of amino acids. When a protein loses its higher order structure, which is a secondary or tertiary structure, but not its primary sequence, it is said to be denatured. Denatured proteins are usually non-functional. For some proteins, the denaturation can be reversed. Since the primary structure of the polypeptide is still intact, which is the amino acids haven't really split up from each other, they are still forming, they are still being held together by the peptide bonds. It may be able to refold into its functional form if it's returned to its normal environment. Other times, the denaturation can be permanent. An example of irreversible protein denaturation is frying an egg. So you're applying heat, which is really breaking down these interactions, that is keeping the albumin protein in its folded form. Those interactions are broken down, the protein gets denatured, but that does not really mean that when you remove the heat or when you switch off the stove, the protein is going to refold into its original form. In this case, the denaturation is irreversible. Which brings us to altered folding or misfolding. Now, protein folding is a very important biological phenomena. Now, just like with everything, things can go wrong with protein folding, in which case the protein is misfolded or partially folded. Now, because a protein has to be in its folded form for it to be functional, whenever a protein is misfolded, it can alter it alters the structure of the protein, which can eventually alter the function. So misfolded proteins can give rise to a number of diseases. When a protein gets misfolded, over a period of time, it starts accumulating. And it is the accumulation of these misfolded proteins that give rise to diseases such as Alzheimer's, Parkinson's, mad cow disease. All of these diseases result from misfolding of particular proteins. Lastly, what is a quaternary structure? A quaternary structure is when you have more than one polypeptide chain held together by non-covalent interactions. The example that you see in this picture is that of hemoglobin. Hemoglobin is a protein that has quaternary structure. Not all proteins need to have quaternary structure. Some proteins that are really big do have quaternary structure. In a quaternary structure, you have individual subunits coming together to form the quaternary structure. So as you know, hemoglobin has 4 subunits. It has 2 alpha subunits, 2 beta subunits. These 4 subunits, each one of them is capable of binding oxygen. It is when these 4 subunits come together to form the hemoglobin molecule, they can bind 4 oxygen atoms. So a fully functional hemoglobin molecule is capable of binding 4 oxygen atoms and carrying it to the rest of the tissues of the body. When something goes wrong with this quaternary structure, it can result in diseases, an example of which is thalassemia. Thalassemia is when either one of these 4 subunits doesn't come across in the right manner to form the quaternary structure of hemoglobin, is when you have inefficient transport of oxygen that results in a disease called thalassemia that we'll talk more about in DDM8. But this is what a quaternary structure of a protein is when you have more than one polypeptide chain that is held together by individual units that can function independently or cooperatively of each other. And that is it about protein structures.